Hello students, welcome to LEELA'S tutorial. So today we will discuss about encapsulation and production of synthetic seeds. Here the synthetic seeds itself implies that these are artificial seeds. If you take in vivo condition, naturally the fertilization or the pollination occurs in flowers where the male and female gametes, that is ovules and the pollen grains fuse to form an embryo which further transforms into or develops into a seed, right? But here, without the involvement of male and female gametes, simply by using the somatic tissue, we are producing the embryoids, which are further developed or modified to form the synthetic seeds, right? So let us see in detail now. So the scientist Murashik is the first person who has prepared or who is successful in producing the artificial seeds in vitro, that is outside the plant body, right? Otherwise you can say in vitro means in the lab condition. Okay, so how we have prepared these artificial seeds? Artificial seed mainly consists of embryoid, which is produced by using plant tissue culture. In last class we have discussed that, right? How you are going to produce or culture the embryos. Okay, so after producing the embryo by using your plant tissue culture techniques, then these are encapsulated or entrapped with a gelatinous substances, right? So as a result it forms a protective coating. So artificial seeds consist of embryoid, artificial endosperm and artificial seed coat. Okay, so what is the difference between the natural seed and the artificial seed? Okay, so here you can see this is the natural seed. Okay, here in the natural seed, this is the embryo. Okay, which results in germination, and here this is the endosperm which provides nutrition to the developing embryo. Then the seed coat, it forms an protective layer or the protective coating, right? Whereas here in case of artificial seed or the synthetic seed which Murashik has prepared, here he has taken the embryoid which is produced by using your somatic cells, right? By using the somatic cells, he has cultured these embryoid in plant tissue culture media. Then he has encapsulated it with artificial endosperm. So this region is the artificial endosperm which provides nutrition to the developing embryoid, right? Then the gel itself forms the artificial seed coat. And here he has selected the carrot plant and he is successful in producing carrot artificial or the synthetic seeds, right? Which were sown later to produce a carrot plantlets, right? So here let us see what are the different types of synthetic seeds. It is of two types. The first one is desiccated artificial seeds. Second one is hydrated artificial seeds, right? So what do you mean by desiccated and hydrated? So I told artificial seeds are entrapped or encapsulated, right? Entrapped or encapsulated, or you can say imprisoned, right, inside the gelatinous substances. So here when these embryoids are entrapped inside the gelatinous substances, these are referred as hydrated artificial seeds, right? So here it is a semi-solid, it consists of moisture, and so these are hydrated seeds. Whereas in case of desiccated seeds, artificial seeds, here you make them to undergo desiccation by placing them in a desiccator, right? All the water content or the gel which is hydrated, right? All the water content or the moisture content will be lost. As a result, the desiccated or shrunken seed will be there. The embryoid will be intact, but here the embryoid will be present, but the gel will shrink or get dried to form a hard coat like structure, which is referred as desiccated seed. This is the hydrated seed which consists of semi-solid or gelatinous substance, whereas hydrated seed only consists of embryoid covered with dried gel. Is that clear? So now these can be used as seeds. You can sow them in vivo and also in vitro. Right? So in vitro, that is in inoculating or sowing them in plant tissue culture media. In vivo, they are naturally sown in soil. Right? So let us see, what is the application? When we have our natural seeds, why we have to go for these production of synthetic seeds? Let us see, the first application is we can propagate many number or different varieties of transgenic plants. What do you mean by transgenic? Yeah, the cells which are introduced with the transgene. The foreign gene of interest is introduced into the cell. Right, and that cell is allowed to propagate to form your embryoid. Right, then that embryoid will result in production of transgenic plants. Is that clear? So it is not that you are introducing the gene of interest into the single cell and you are getting only single plantlet. You will be introducing the gene of interest into the single somatic cell and you are allowing it to divide so that a ball like cells are formed. That is nothing but your callus. So from the callus, the hundreds of plantlets can be produced, which can be transgenic plants. Is that clear? So, and the second application, it is you can produce the plantlets or you can select the somatic cells from the plants which doesn't have any seeds. So, non-seed producing plants, somatic cells can be taken and you can produce the synthetic seeds. Is that clear? Then in case of seed propagation problem, in some seeds there may be problem in germination due to the very hard and thick seed coat. Right? Otherwise there may be prolonged dormancy, seed dormancy may be there. So in that case also, instead of using those natural seeds, you can go for this artificial or the synthetic seeds. Is that clear? So these are the main applications why we are producing the artificial seeds or the synthetic seeds. So let's see in detail now how you are going to produce. So first you are going to select the plant. See, either if you want to produce a transgenic or you can select the plant which doesn't have seeds or you can select the plants which have seed dormancy. Right? So select one plant like that. Then you are going to take the somatic cell. See, unlike in natural process, we are not making use of any ovules or the pollen grains. There is no fertilization here. We are producing the embryoids by using somatic cells. What is difference between somatic cells and the gametic cells? Gametic cells are your ovules and the pollen grains. Whereas somatic cells, other than these two cells, right? Other than the gametic cells, the complete plant body consists of somatic cells. So any tissue you can take, right? So any tissue, select any tissue from the plant apart from ovules and the pollen grains. Then you just culture them in plant tissue culture media so that you get a callus. Right? If you want to produce a transgenic plant, what you do? You select the cells. Right? You select the plant, select the explant, then cell suspension culture. The single cells will be introduced with the transgene. Then these genes, these cells which consist of transgene will be allowed to produce a callus. Right? So that callus in the plant tissue culture lab, now once the callus is ready, by means of your auxin to cytokinin ratio by variation, so what happens here? The meristemoids are formed over the callus. So here these small spikes like structures are formed over the callus. This is nothing but induction of somatic embryoids. Right? Or you can say induction of somatic embryogenesis. Right? The embryoids are generated from the callus. So you get so many embryoids from single callus, segregate them, and then here you can see completely developed somatic embryo. Why you are calling it as somatic embryo or embryoid? Right? No, natural embryo is developed from two haploid gametes fusion to form a diploid embryo. Whereas here the embryo is formed from somatic cell itself. The diploid cells are producing the diploid embryo. So it is referred as somatic embryo. See here, one more thing. Here we are calling this as embryo, but when you are producing in the synthetic seed, you are calling it as embryoid. Is that clear? So, embryo is naturally occurring embryo, whereas here, if it is an artificially produced embryo, we call them as embryoids. Right? Embryoids or somatic embryo. Okay. So, here once you get the somatic embryo, what you are doing? You are segregating all the somatic embryos and then you are preparing 0.3 to 5%. That means 0.3 grams of sodium alginate dissolved in 100 ml of distilled water. So, that solution you are going to prepare and you are going to drop all the somatic embryos. Everything must be done in laminar airflow under sterilized conditions. Right? So, now you are dropping them in sodium alginate solution. Then make 100 ml of calcium chloride solution. Right? So, calcium chloride solution, you are going to take it in a beaker. Then you are going to place a magnetic bead. Right? Magnetic bead is placed inside the calcium chloride solution and you are going to place this beaker over magnetic stirrer. So, the magnetic stirrer will be there. Okay. So, once you make everything, all the setup ready, what you do? Take the syringe or the dropper and suck some of the sodium alginate solution along with the embryoids. Right? So, here you can see the syringe consists of sodium alginate solution consisting of your somatic embryoids. Then, now pour it drop by drop. Right? Drop by drop, when you are dropping it, here the magnetic stirrer will be on. When it is on, the magnet present in the magnetic stirrer start rotating. So, very gentle rotations like 20 to 50 RPM, that is rotations per minute. Right? So, when it is rotating gently, the magnetic bead which is present in the calcium chloride solution will also start moving. Right? That means you are simply swirling the solution. Okay? So, you are simply swirling the solution, but there should not be any vortex formation. Right? So, simply swirling the solution, you are pouring this drop by drop. So, because of that, see here, as the drop is falling into the calcium chloride solution, they are forming a gel-like beads. This is because of, here we have taken sodium alginate, here we have calcium chloride. As sodium alginate is dropped in calcium chloride, this is forming what? Calcium alginate beads are formed. Is that clear? Calcium, when alginate is touching to the calcium, due to the reaction, here what is forming? You are forming calcium alginate gelatinous beads. So, during this gelatinous bead formation, your embryoid is entrapped inside these beads. Is that clear? Entrapped or encapsulated or imprisoned inside. Okay. So, here what you do, in this sodium alginate itself, you will be pouring certain nutrient substances so that they will act as your artificial endosperm to nourish the endosperm. Is that clear? So, when the bead is formed, now this bead consists of embryoid and the sodium alginate gel consists of certain nutrients which provide nutrition to the embryoid. Right? And the gel itself acts as the seed coat. Now, after this, when you separate them, okay, so now this process is called as calcium alginate encapsulation process. Right? So, after encapsulating the embryoids, now either you hydrate them, otherwise you desiccate them. Hydrated means you are maintaining them in gelatinous structures intact without allowing them to dry. Right? In case of desiccated, what you do? You allow them to dry in desiccator so that all the content or all the water content present in the gel will be evaporated so that you get a dried seed-like structure. Okay, so now desiccated or the hydrated seeds can be used in vivo or in vitro. Okay, so here I am showing you in vitro condition where we are sowing in lab. Okay, in the plant tissue culture media. So here you can see in the Petri plate you have the plant tissue culture media where we have sown the hydrated seeds. hydrated artificial seeds, whereas here we have inoculated the desiccated artificial seeds. In both the cases, here the germination occurs and they result in formation of plantlets. Right? So after inoculation and incubation, here the germination starts. After few weeks, you can see the formation of root and shoot system. In both the cases, in case of hydrated and also in case of desiccated. And these, after formation of the plantlets, the complete organs are formed, the shoot and the root. Now you can transfer them to the pots. And then further you can transfer them to the greenhouse and later to the fields. Is that clear? This is the use of synthetic seeds. If you have any doubts, put it in comments and subscribe for further videos.